Hi, my name is Katie, and I will be presenting the role of beer lipid-binding proteins in preventing lipid destabilization of foam. The question that this paper asks is, how do fatty acids, either long or short chains, affect the foam stability of the beer? And this is very important because foam and flavor are basically what makes beer taste good. But there are lipids that can be introduced into the beer or lipids that are already present in the beer that could damage the foam. The paper began by doing hydrophobic interaction chromatography. The steps include to pre-equilibrate your column, load, wash, and elute. And they collected 5 groups, group 1 being the least hydrophobic, and group 5 being the most hydrophobic. After this, they continued to do SDS page, and the results shown here are to the right. And it showed that there is a 40 kilodalton broad smeared band here, and also group 5, which is shown here to the right, had a molecular weight band that was higher than the rest of them, and that's circled. Next, they continued on to fatty acid binding. There were 2 types, C14 labeled linoleic acid and fluorescent lipid cis perinaric acid. And there were some basic steps that a stock solution was prepared, and for the C14 labeled linoleic acid, the stock solution was diluted. Aliquots were taken for LSC, which is a liquid scintillation counter, which counts how much radioactivity is in that aliquot. And then 90 microliters of your sample was added to a column. And afterwards, it's basically like the hydrophobic interaction chromatography, and then they collected the sample after the rinse, and then they counted it by LSC to see how much counts are in that ending eluate. Now for fluorescent lipid cis perinaric acid, a stock solution was prepared, and then the group 5 and group 1 and BSA, which is standard, were titrated by adding 5 microliter aliquots of CPA. Here are the resulting fatty acid binding assays, and you can see in the figure 5 below that the binding curves show that group 1 and group 5 have very shallow binding curves. Group 5 was saturated at about 4 micromolar, and group 1 was saturated at about 5.5 micromolar. And BSA, which is in the top right corner, had a far greater fluorescent response and a steeper initial slope than either group 1 or group 5, and it became saturated at about 6 micromolar CPA. The KAs were, or the associated constants, were calculated, and group 1's Ka was 6.27 times 10 to the 7th, and group 5 was 1.62 times 10 to the 7th. The foam stability was also measured from this paper, and they measured it using the radiometer Copenhagen CDM83. Basically, a sample of beer was added and nitrogen was forced through a jet, and it created a foam up to 30 millimeters, and then the foam conductivity was measured. The foam stability results show that when group 5 was added to the control beer, a small enhancement of the foam stability occurred, and when group 5 was also added to a lipid damaging beer, it significantly restored the foam. Overall, the main conclusion was that long-chain fatty acids have more of a negative effect than short-chain fatty acids.